I'm going to go ahead and introduce Katja Weinacht. Dr. Weinacht is an assistant professor of pediatrics and a pediatric hematology oncologist with a special interest in diseases that intersect immune dysfunction, primary immunodeficiency, and bone marrow failure. Her clinical practice focuses on pediatric patients requiring a hematopoietic stem cell transplantation, patients with DiGeorge syndrome, and patients with genetic immune diseases presenting with autoimmunity. After receiving her MD, PhD degrees from Technical University in Munich, Germany, she completed her postdoctoral clinical and research training in a clinical fellowship at Boston Children's and the Dana-Farber Cancer Institute. She did a research fellowship at the Harvard Stem Cell Institute, where she acquired skills in the field of reprogramming, tissue engineering, and gene correction. And I've had the pleasure of collaborating with Dr. Weinacht related to understanding thymus development and its implications for DiGeorge syndrome. So I'm very excited to present. The next several talks we are having are focusing on fetal development. And so this will be the first of those. And then the later session will be focusing more on placenta, pregnancy, and uterine tissue. So with that, I'm going to hand it over to Dr. Weinacht.
Thank you so much, Virginia. I'm excited to be here. And what most of you don't even know, and maybe Virginia doesn't, and one of our next speakers, Vittorio, doesn't, the only reason why I am at Stanford is probably because of this group, and we will cover this in this talk. So when you asked, do I want to present? I said, yes, absolutely. This is my way to say thank you. How important maternal-fetal medicine is and how profoundly these human tissues can influence what quote unquote unrelated people do is really beyond expectation. So how do human fetal tissues inform our in vitro generation of thymic epithelium is what I'm going to talk about. And you see how excited I am to talk about this. Let's see if I'm able to navigate this as well. Yes. Okay. So briefly, I'm a stem cell transplanter. I'm an immunologist at heart, and I think a lot about the thymus. And so what does the thymus do? You know, all know, yeah, sure, we are clinicians. We know about the thymus. Relatively little, let me pause it. So the thymus is a squishy organ in the center of your chest. It peaks in size in infancy, and by the time you get your driver's license, it's essentially structurally undetectable. And in the thymus, we distinguish a cortex and a medulla. Accordingly, we distinguish cortical and medullary thymic epithelium, and that's we see right here. Dark is the cortex, light is the medulla. It has more nuclei, and this is why it stains darker. And what I'm highlighting here on this beautiful scanning. If you're going to step away, let's get your mic. Yeah, that's good. So I am known to move around. No. But you can hear me in this room, right? Yes. All right, let's put this somewhere. Can we get confirmation that the Zoom people are hearing? Can the Zoom people hear? Okay, Zoom people can hear. Now you hear more than you ever wanted to hear. Let's talk about the thymus. So in the thymus, we have two types of cells. We have the thymic epithelial cells, and then we have the small, round thymocytes, which are the cells of hematopoietic origin, which are T cell precursors. So when we ask the question, what does the thymus do? There are mostly 3 to 4 things. A, thymic epithelium attracts common lymphoid progenitors into the thymus. It makes them multiply, it makes them mature, and then most importantly, it selects a broad and self-tolerant T cell receptor repertoire. And let's think about this just for 1 minute, and I promise I'll stay on time. So 500 billion to 1 trillion T cells percolate throughout your body the entire time, and they see the world through the T cell receptor, and they touch a peptide in the context of HLA, and they say, self, you stay, self, you say, self, you stay, foreign antigen, you go, and and the T cell destroys the cells. That's what a T killer cell does, right? And so how does this T cell receptor repertoire actually come about? That is a very important question, because T cells make all random types of T cell receptors, and then 99.9% of these cells die. So this is a thymic epithelium. This is a bacteria, virus, cancer cell. And the T cell comes, and the T cell receptor engages with the thymic epithelial cell. And based on intermediate affinity to self-antigens, it is projected that you have strong affinity to all these bad antigens, and you get survival signals, and you stay, and you give rise to a broad and self-tolerant T cell receptor repertoire. Now, next T cell comes, has high affinity to self-antigens. Danger for autoimmunity, you need to be clonally deleted. Off you go. And then there is a third type of, this very important function of deleting autoreactive T cell is redundantly executed by dendritic cells and macrophages. Very important when we speak about tolerance later on. And then there is a very special type of the thymic epithelial cell in the context of AIR, autoimmune regulator. That's the one that nurses regulatory T cells. And then the T cell receptors that don't bind, they die by neglect. So let's say we made 1 million T cells, most of them are bad, one is good, and that stays and that goes in the circulation. Now, imagine what happens if you don't have a thymus. If you don't have a thymus, you clearly make no T cells, right? You have no way to make them, although your hematopoietic system is normal. And you present like this is the quintessential David Fetal. This is the child with SCID, with severe combined immunodeficiency, the bubble boy disease. He had a hematopoietic problem. And so we could cure him with a hematopoietic stem cell transplant. And the worst thing is when we misdiagnose patients for having a hematopoietic problem, they have no T cells. We think they have SCID. We transplant them. They still have SCID, and then we know they have a thymic defect, and then we are in deep, deep trouble. Now, what it would be theoretically the therapy of congenital athymia, of not having a thymus, a very visionary woman at Duke, Louise Marquardt, like back over 30 years ago, suggested, well, we can give thymus from a donor and we transplant it under the knee, literally, of the recipient patient. Minor issue, that tissue is not matched, because where do we find thymus? We find thymus, it's under the breastbone. It's only available in an infant. It's when Casey just consented somebody for, a study who's about to undergo cardiac surgery. These will never be matched. We are transplanting HLA unmatched thymic tissues. So we are creating a bomb in that patient, or what? Well, positive selection comes from intermediate affinity of self-antigens. And if you are intermediate to myself or you are intermediate to Virginia, hopefully you still recognize a cancer cell as foreign, and that's okay. And then negative selection, luckily, is redundantly from this dendritic cell, and we are all okay. But what we are missing is that thymic epithelial cell that makes autoimmune regulator. And so these patients that are recipients of thymic transplant, and one of my patients is just here, was briefly in the ICU with you, from wicked autoimmunity. And so we really need to do better. Okay. And so now every good story starts with a visionary protagonist. And here is our visionary protagonist, the first person, and probably the reason why I ultimately came. Maria Gracia recruited me to work on this project. And I said, I have no idea about developmental biology, but Vittorio was there and says, we can do this. And so here we did. And what did we dream up? We dreamed up the following. What if we take induced pluripotent stem cells, we turn them into thymic epithelial cells, we give them back to the patient, and wonderful, we have a diverse self-tolerant T cell receptor repertoire. Minor issue. If you have congenital athymia and your body can't make cells that give rise to thymic epithelial cells, probably in the dish we can't do it too, right? There's a cell intrinsic defect. But here I'm a stem cell transplanter. I talk to the National Donor Matching Program. We can, of course, get blood that is HLA matched. We make the iPS cell from it and we give that to the patient, and that actually works. So great. We spoke to the National Donor Matching Program and they were extremely excited about it. And not only has this implications, curative implications for our patients with congenital athymia, patients who had a bone marrow transplant. Now we're talking about my peer group, and we should not tap on the microphone by the same time. These patients have thymic injury from irradiation, from infection, from graft versus host disease. The thymus is, of course, MHC rich. This is the first where graft versus host disease attacks. We just don't see it, except for these patients later have no T cell reconstitutions. they would be recipients of that. And then last but not least, for the cardiologists and cardiotransplanters in the room, we could of course make thymic epithelium match to donor organs, vastly expanding the donor pool, and therefore, it really transplant solid organs safely without lifelong immunosuppression and the morbidity with it. So it would be really cool. And Vittorio says, no problem, we can all do this. We go down endoderm, foregut, third pharyngeal pouch, thymus, and we block going to midgut, hindgut, bronchus, lung, and parathyroid. So we thought this is easy breezy, we're gonna do it. And here's what we did. IPS, definitive endoderm, anterior foregut, all these stages were well known. And then the last three here, they were unknown, but we had each other and we thought we can do this, no problem. And we took whatever we did in the dish, our iPS cells, and we transplanted them in an athymic NSG FOXN1 mouse that is an NSG mouse, a mouse that is permissible of human cell engraftment because she has a hematopoietic problem. She does not have her own T cells. She doesn't reject. And then we gave her a biallelic FOXN1 mutation that is the key transcription factor for thymic development. So she has no immune system and she also has no thymus. So she's the perfect recipient to test this. We give her our cells and here's what we get. We get these bubbles and we're like, does it look like thymus? Not so much. We analyzed it, and by all the developmental people in the room to whom NKX 2.5, SOX2, PXUJ1 and others have a lot of meaning, we compared this to fetal bronchus and fetal lung, and that's what we did. We make respiratory endothelial cysts. Now, interesting piece of information, we made these cysts mice that do not make a thymus, actually that have this knockout mutation in FOXN1, they in and by themselves make these respiratory cysts. And the idea is that FOXN1 overrides respiratory cell fate as the default developmental program in the anterior foregut. And then even more importantly for the stem cell transplanter, a regular NSG mouse who does have a thymus and makes lousy but some T cells also has this cysts, meaning that FOXN1 is necessary but not sufficient to fully suppress that respiratory epithelial cell fate bias that these cells have. So, and then the person who saved us is right here. So after Vittorio, the next saver is Virginia. We went to Virginia and we said, okay, we need human fetal tissue of bronchus, lung, esophagus, also the anterior foregut, and then the parathyroid that actually is even closer related to the thymus because they both branch off of the anterior-most portion of the anterior foregut, the third pharyngeal pouch. So you go right, you go to thymus, you go left in the anterior pouch, you go to the parathyroid. So we looked at all these organs at the fetal stage, all human. We called this parallel, and then orthogonally, we also looked at human embryonic, fetal, and postnatal thymus. So we had both these spreads. And then we did a lot of sequencing, integration, and then an absolutely wonderful undergraduate student, the most brilliant undergraduate student I've ever worked with, Mohammed, came and said, okay, I'll do this, I'll take this on. After a while, and so the first thing we did was we employed an algorithm called CNIC. And what CNIC does, and you have to be very careful when you deal with this large-scale data sets, it's an algorithm that looks at developmentally important transcription factors, pairs them to their downstream target that have a validated consensus sequence. So as far as large-scale transcriptional data goes, it's as much validated as we possibly could. And when we just do a UMAP, compressing multidimensional data into 2 dimensions, we see that the thymus clusters apart from all of this, very beautiful, sort of expected. And then we asked the next question, what are the transcription factor regulons that actually stand out? Then we found that out of these thymic regulons, number A was the one in black, and the black one is here, is totally high in the thymus. All of these, and there were many more hidden in these data pods, they're all related to interferon response genes and to NF-kappa B. And nobody had ever described this before. Interferon response, NF-kappa B, we thought, is there a glitch in the algorithm? So we applied multiple different ways to look at it in different ways. We built very stringent modules that looked at interferon response. And no matter how we sliced it, in the cortical thymic epithelium, in the medullary thymic epithelium, interferon was always high. Everywhere else it was always low. We even looked super early in development, week 6, with the parathyroid thymus was high, parathyroid was low. Then we looked sequentially over time, week 6, week 7, then here the fetal stage, and then postnatally. And we saw that interferon signature only going up. And everybody who's a little bit an immunologist will say, well, the thymus is full of thymocytes of T cells. They give the interferon, sure. So then we stained for interferon. And this was a beautiful confocal, but it doesn't show up very well. All this is thymus, cortex, medulla. This is the corticomedullary junction. And in essence, everything shows up. Pancase, the thymic epithelium. But you have not only the small cells that are the T cells show up, even the epithelial cells. And then a big debate in my lab spurred and said, well, you don't know whether the interferon actually comes from the thymocytes or from the epithelium itself. And I said, of course, I'm an immunologist. So in the end, we settled on, we sort the cells, we do bulk sorting because single cell is not fine enough to pick up interferon transcripts because it's such a lowly expressed transcript. So the entire lab got together, everybody had to isolate the cells, it was a big schlep. We did bulk sequencing and clinical gestalt is sometimes right. It was the thymocytes, so the pre-T cells that were the suppliers of interferon. So that was settled, finally, peace was in the lab again. And guess what we did? Of course, in our last step, we said, okay, how about we sprinkle some interferon in our differentiation, and from there on in, things started to make sense. So when you look at definitive endoderm, anterior foregut, third pharyngeal pouch, then we ventralize, we go right to the thymus, left we would go to the parathyroid. All these transcriptional patterns, they made perfect sense. We validated them on a protein basis. We also demonstrated that up to 92% of our cells were EpChem, an epithelial marker, and HLA-DR, an antigen-presenting marker positive, and that is a hallmark of the thymic epithelium. Then, as of yesterday, we got single-cell RNA sequencing data back. And Casey, just so you know, this is just the scout. This is only a 70th of the sequencing depth. The real images will look so much prettier, and yet we were dancing. EpChem, epithelial marker, all our keratins are there. FOXN1, imagine this 17 times higher, is there. HLA-DR, all our key markers. And this is a marker that is white cell dependent. It only comes up when lymphocytes come. So our interferon actually does something. And again, this is just the scout images. So we were absolutely in heaven. It also started to become fun once we knew what we were doing, is usually the case. The cells really develop like a 3D structure, and you see how they develop. They really all of a sudden start to have their own life. And of course, the thymus is a 3D epithelium and has never ever before been derived from embryonic stem cells or iPS. It's one of these niche organs that could never really be developed. So we partnered with Sarah Heilshorn from engineering to get the right stiffness and to get the right biomatrix, because you don't want it too stiff. You want thymocytes to be able to enter in. You see, if you do regular martigil, it's like rock hard, way too stiff. When you add laminin, they're like, da-da. They're very happy with that, obviously. And then we did a first sort of cheap women's experiments where we transplanted them into nude mice. Nude mice are not NSG mice. They can reject cells. They just do not have a thymus. They have a little bit of spontaneous T cell development, but we were excited. We see T cells going up. This is the kidney capsule, and this is the graft. And when you look at the graft areas here, there was clearly T cells and clearly T cells here. So we were quite excited that this would work. But of course, we got more ambitious and we said, oh, let's do this in the gold standard model. Shouldn't be that hard. Okay, so the gold standard model would be a humanized NSG mouse. So it's a humanized NSG mouse. She has no immune system of her own. She will not interfere. She will not make her own T cells, certainly not spontaneously, because she can't. And if we give this human mouse now a bone marrow transplant, then human HSCs go to her thymus. And in her thymus, there will be T cell development. As I told you earlier, it's not perfect because her thymus has some cysts, but it's okay. She will make some T cells. Now we give her a biallelic mutation in FOXN1, and she becomes an NSG nude mouse. She's now permissible of human cell engraftment, and she doesn't have a thymus. And if we give her a stem cell transplant, and then look at T cell development, there is nothing. And so now the gold standard of testing of our cells actually do what we claim to do is if we put functional thymus, primary or iPS derived under the kidney capsule, and then she makes T cells. And so this is how, this is the kidney capsule when we transplant. This is right after transplantation, we see nothing. This is the contralateral kidney, and then this is this graft. This is actually in a nude mouse. This is a very small graft. And so we did this first as a pilot in a small number of mice, and okay, my slides are not in order. And all of a sudden, oh my God, what we saw was that, let me briefly slump ahead. What we saw in these two mice is that now we had huge grafts and we had this extremely high T cell numbers, catered to the idea that now we had the perfect model to do that. Unfortunately, then the mouse pandemic hit, and now I'm going back. Unfortunately, then the mouse pandemic hit, and for 18 months, all our mice died. The story ends well, don't worry. And then finally now with the mouse pandemic, we're able to breed mice again. So we partnered in that time with the laboratory of Agnieszka Czekovic and a wonderful technician who's Leah Schwarzrock, who helped us establish and transplant these mice in a much safer way using CD117 saporin conjugated antibodies, because this antibody conditioning is not that harsh on the mice and the mice can actually survive. And what you see in our negative control, we give them a human stem cell transplant. They make human CD45 cells, human white cells. They make absolutely no T cells, but they make B cells and they make myeloid cells. And then as a positive control, if we give this mouse a primary human thymus, then oh yeah, for sure, we make CD3 cells, and they have a naive phenotype. That's really what we're looking for. So it's CD45 RA positive, and it is RA and CCR7 positive. So there are new T cells being made. Very excited about this. And then we do this in our mice, and oh my goodness, our mice was a super maker of T cells. You can see how thick these plots are. Interestingly enough, they have a, I'm looking myself again here, they have a little bit less naive T cells than the primary tissue. And we think that although our thymic epithelium is perfect, the glycosylation is still not, I mean, we've done this for the first time. They're not as perfect as wonderfully naive human T cells, but they are T cells and they do work. And we know they do work because they give the mouse xeno-GVHD. And now we have 30 more mice coming down the road and testing them in different hydrogels. Now, what I want you to see is really how these structurally look. So primary thymic tissue here, hallmark of thymic architecture is these Hassel's bodies, and we think they are T cell graveyards, if you will. Now, this is an NSG mouse transplanted, and you see in the NSG mouse, we have this massive expansion of white cells. These are all white cells in the tissue. Here you see these, well, you see these islands of white cells. And we even found Hassel's bodies. This is in an iPS-derived graft. So there's really, it is the same type of physiology that we see is going on. And if you now compare, this is primary human fetal tissue, you're looking at CD3, so you're looking at T cells and pankeratin. You're looking at PAX9, one of the key transcription factors of the thymus. And this is, of course, zoomed into a small fraction. These pictures look virtually indistinguishable in the small areas. You see T cells, you see the keratin carrying thymic epithelium. You see PAX9 as one of the key transcription factors. I show you two more, and then we are already done with the talk. PSMB11, one of the hallmarks of the thymus, PSMB11 is the proteasome that processes all these peptides that are then presented on the surface of the thymus. We see wonderfully in both. And here I'm showing you sort of the overall architecture. So this is the mouse kidney. These are all the nuclei. This is the pankeratin. So what you see is that our thymic grafts, they have these islands of thymic epithelium. And around these islands of thymic epithelium, this is a very specific marker for the thymus. Then there's also the T cells really clustered to it, exactly what happens in the thymus. So of course, we're very excited about these data, and we are in the process of trying to refine this as a therapy for patients, really. So steps that will be next is how can we get rid of these other cells? There's small clusters of neuronal cells and other clusters of stromal cells, and how can we purge the cells either through sorting, through getting them through a column, and then immediately packing them into an organoid and transplanting into the mice. So I hope in the past few minutes I have convinced you that really, if it wasn't through partnering with your center, Virginia, I'm standing right in front of you, if it wasn't partnering through you and having access to human fetal tissues, we would have never figured this out. The best organoid labs in the entire world have tried to make thymus, and only because we analyzed the lung, the bronchus, and the esophagus to the thymus in parallel, we could figure out that it's actually interferon. Who would otherwise ever know? Nobody ever went even in that direction at all. So thank you so much for this collaboration. It's really exhilarating. We are quite excited about it. And the fact that this is functional and we want to take it to the patients. And Vittorio, I think you are just up here. I spoke about you earlier. I said every good story starts with a good protagonist, and here he is. And with that, I'm at the end of my talk and happy to take any questions.
Great, thank you. Any questions that came up through chat? No. Okay, great.
Okay.
So
There's a question right here.
I'll ask a question just because I can. Do you know whether the cells that you're producing are more cortical or medullary in their phenotype?
Yeah, so that's why so again, these are just the lame scout images on the single cell RNA-seq. And again, there's 70 more data coming, but I was just not ready in time for this talk. And my lab actually was unhappy that I showed the dirty images. Well, so when you look here, clear markers of cortical text, CCL25 is a clear marker. FOXN1 expression in C-text is the highest right here, and PRES16 is a downstream target, and still it is necessary to have thymocytes present. So this is really your C-tech cluster, and then your who do we have? And here, CCL19 is a little bit wider and is almost everywhere. And there's these intertypical texts that are called MC texts that have low expression of both. And this is what we see here. We've just have a paper with Virginia under review that looks at all these different primary cell populations from the data set that I've just been showing you. And so we think, and we are actually quite excited that we have these intertypical early markers. What I didn't show, there's also a cluster here of cycling texts, and the cycling texts are coming even before these intertypical texts. So right now we do have a C-tech bias. And what we see, I didn't go into so much detail. If you go down the NF-kappa B route, you go more towards C-tex. If you push the interferon higher, you go more towards the N-tech route. And our delivery of interferon, although we use it in really, really high doses, is limited by the fact that we have a lot of extracellular matrix at this stage. So what we're doing right now is that we are moving the interferon at even earlier stages when there's less extracellular matrix, and then we need to do another round of single cell to see if that creates more of an M-tech bias. And then to actually do M-tech2 is probably the next question. How do you get air in there? That's one of the things where I would say we CRISPR it in there.
A quick follow-up. Does this data set then, or in your opinion, even beyond the data, speak to the question of whether there's a common progenitor
for
shared for the cortical and medullary human as in mice?
I can't speak to the mice, but I look at the camera wherever Vittorio is. Every question of the mouse, I immediately defer to Vittorio. But as far as know for the human, and we've looked extensively, there's not a lot of stem cell markers, even in primary thymus. And these very early samples, Virginia knows we got them through the UK because their means of abortion is slightly different. So week 6, week 7 embryos we even looked at, and we see more of these cycling texts, and we think that cycling texts are the ones that very early cause an expansion and then give rise to these intertypical MC texts. And why the thymus doesn't last, we personally think, and this is the next frontier of my laboratory, is that interferon has something to do with it. We all know that in the hematopoietic compartment, right, the hematopoietic cells, they exhaust in the context of interferon. And now here you have an organ that is showered in interferon. Probably nobody can survive for so long with such an interferon shock. I mean, that's really what I think where all of our research will be going. Yeah.
Thank you.
Remind me again, so thinking back to the DeGeorge, which I think was some of the inspiration.
Yes, exactly right. It was these patients that we couldn't cure by bone marrow transplant.
Yeah. So the in the DeGeorge, do they need the what is the replacement that might work?
The exact cell that we are making, the thymic epithelial cell, that's the cell they need.
Yeah.
And
so then the question would be, at what point of fetal development? So is this the type of thing
that needed to go in utero
versus neonatal?
I think this could totally be neonatal, because in utero you're relatively sheltered. And then you come out and you still have lots of maternal antibodies. You're still fine, and then your mom breastfeeds you and you get CMV, and this is when things go down. And so the lifespan of an untransplanted patient with complete DeGeorge, with complete thymic aplasia, is only 1 to 2 years, even if the best preventative measures. And the lifespan with a patient who gets a thymic transplant is no longer limited by infection, but is limited by life-threatening autoimmune. And we are caring for one. I'm actually on service. I'm walking back there to that one right now. And of course, then Casey and I have another project where we think, instead of going HLA matched, can we go and can we rescue? There's of course much more than interferon. One of it is retinoic acid, and this is probably all Vittorio is going to talk about. Can we rescue retinoic acid metabolism?
Great.
Thank you so much.
Any other questions? All right, let's give Katja another welcome. Thank you for her outstanding and inspiring talk. So next up, we're running a little ahead of schedule, but I think we'll keep on moving.
Sitting with my friend now.
Next up, we have another remote speaker joining us from across the Atlantic, I think. This is our very own Dr. Sebastiano, who's an associate professor in the Department of Obstetrics and Gynecology, and he's been named the Woods Faculty Scholar in Pediatric Translational Medicine by the Stanford Maternal and Child Health Research Institute. He received his PhD in cell biology from the University of Pavia and completed postdoctoral training in developmental biology at the Max Planck Institute for Molecular Biomedicine and in regenerative medicine here at Stanford School of Medicine. He's an expert in stem cell biology and epigenetic reprogramming and has contributed significantly to the understanding of our somatic reprogramming using induced pluripotent stem cells. Notably, his lab has pioneered new concepts in regenerative medicine and for the treatment of aging-associated diseases by repurposing the principles of reproductive biology and applying them to somatic cells. Today, he will share with us his work on in vitro generation of germ cells as we continue our theme of fetal development. So I'll turn it over to you, Vittorio.
Thank you so much, Virginia. Can you hear me well? You can hear me?
Yeah.
Okay, great. Yeah, thank you so much for the introduction. Hi from Barcelona, where I'm currently based. And also thanks to Katja, who gave also a very nice introduction to the work that that we've developed together a while back. Yeah, so I'm very excited to be here today and to tell you about one of the research projects in my lab. Now, at the very high level, what we do in the lab is really to try to repurpose in a way the principles of embryonic development, human embryonic development and germ cells development, for the goal of kind of treating aging and extend the health span of people. And I have a particular interest in women's longevity and women's reproduction. Now, we do this in two ways. The first way is by stimulating the endogenous cells and rejuvenating the endogenous cells using a method that we developed in my lab, which is called ERA. And the second method is by generating new tissues. And I have a particular interest, in the thymus. The work was, nicely summarized by Katja, at least, the very beginning of it, and the ovarian tissue regeneration. Sorry, let me just move this on the side. Okay, but today I will be talking about basically the work that we are doing in the context of gonadal ovarian tissue regeneration, and how the potential implications of this into women's health span, extension, longevity, and reproduction, of course. So why am I connecting reproductive ability and aging? Well, there is actually a lot of literature suggesting that there is really a clear and close connection between reproductive capacity and aging or longevity. For example, if we look at animal models, we see that if we ovariectomize mice, and ovariectomy is a crude way to mimic menopause. It's not exactly the same thing, but it's, in a way, it's a model of menopause. We see that the ovariectomy basically leads to shortening of lifespan and reduced health span in female mice. And it affects a variety of different organs and systems. It's impacting cognitive ability, it's impacting frailty. If we look at, in women, we see that the time of menopause onset is linked to the susceptibility of age-related morbidity and mortality. And I'm obviously talking about women that enter menopause very early in life, so women that are affected by POI or POF. But there is also a link in women that enter normally in menopause. And there is a Dutch study that, it's not a huge number of individuals, but it clearly shows that for every 1 year of increase in menopause age, there is an age-adjusted mortality rate that is decreased by 2%. So what I'm trying to say here is that there seems to be an anti-correlation between the ovariectomy and menopause and the kind of the morbidity and the mortality that you see with aging in animal models and in women. And what's really also very exciting is to see that if we go back to mice, if we now take reproductive aged animals, you ovariectomize those animals and you transplant young ovaries, there is an increase in the lifespan of these mice. There is reduced inflammation, improved cognitive function, and there is also physical performance of the recipient is enhanced, suggesting that a young ovary put in an old host seems to impact healthy longevity in a very significant way. So if we take all this together, there's definitely a lot of information to suggest that if we could potentially rejuvenate the ovary, this could have an impact on the health span and the lifespan of women. And so one of the goals that we have in the lab is really the question, if you wish, and the goal is to really try to prevent or even reverse the accelerated aging that is triggered by early menopause by rejuvenating the ovarian tissue. And by so, we hope and we think that we could impact women's health span and lifespan. And so how do we do this? Well, we do this in 2 different ways, or at least I'm thinking about this in 2 different ways. The first way is by using the ERA technology, which is what I described before. I will not have time to talk about this today, but we have published the work in 2020. And the basic idea here is that we can, by controlling the reprogramming process, we can basically turn any human somatic cell into its young counterpart without affecting its identity. And so we are also thinking about using the same strategy to rejuvenate the ovary in situ, in the body, and see how this rejuvenation impacts systemically the body. The second strategy, which is the one that I'm going to talk to a little bit today about, is the ex novo generation of new, fresh ovarian tissues from stem cells. And the idea, again, we're far from it, but the general idea would be that if we could generate young new ovarian tissues, which is of course immunocompatible, following somehow what has already been beautifully described by Katja. We could basically potentially think about transplanting these new ovarian tissues into the host. And since the ovarian tissue would be very young, very youthful at that point, we could again potentially impact the health span and the lifespan of postmenopausal women in their early, women affected by POI and POF. Okay. So obviously it's a very challenging, similar to the thymic epithelial cells development. It's a very challenging task for whatever reason. We keep picking very challenging tasks. And in the case of germ cells, it's a challenging task because the germ cells development happens in different parts of the developing embryo. And it's divided roughly in 3 phases. There is a first phase, which is called induction, that happens very early, during development, soon after the blastocyst implants into the uterine tissue, and is basically characterized by this formation and also the early migration towards the prospective gonadal tissues. The second phase is the migration and the colonization of the gonadal tissue, which are, roughly positioned in the back dorsal side of the embryo. And that's where basically the cells become fully committed to the germ cell fate and proliferate heavily. And then there is the third part, which is sexually dimorphic. It happens in the gonadal tissue, in the fetal tissue during female development, and it happens postnatally in the testis during male development, which is the meiotic maturation. Now, it's obviously very challenging. Try to recapitulate in vitro this whole development is very challenging. And so today I will just summarize our efforts in try to really robustly get to this first stage of development, which is the induction and early migratory stage of development of germ cells. Now, I have to say that in mouse, this has already been completely tackled. So both fully developed sperm and eggs have been developed in the case of mouse embryonic stem cells. But the same thing is not true in human, where the generation of germ cells has been far less successful. And this is obviously due to a variety of reasons. Well, first of all, because human and mouse development, as you can see here on the left side, are very different. There are some conserved basic mechanisms, but the specifics of development are quite different. And the only successful attempts so far that you can see here, done by the labs of Surani, Saito, and Amanda Clark at UCLA, have been done by protocols that are not fully defined. And protocols that are still relying on 3D organoids, which are a great tool for discovery, but prevent some fundamental mechanistic kind of studies to be done. And there has been one great example of generation of human oogonia done by the lab of Mitinori Saito in Japan. But the protocol is still kind of relying on the mixture of human and mouse tissues, and a full human-based protocol of development hasn't been successfully done. And so one of the things that I think, or one of the limitation factors that I think is preventing this successful development is, as I said, the fact that the protocols are still, I would say most of them, if not all of them, are still relying on 3D organoids. And so the reason why I think this is impacting the whole process is that if you take 3D organoids, although again, as I said, they're great tools for discovery and development, the fact is that any growth factor in the media somehow has a gradient of accessibility inside the organoids. And so if you are dealing with organoids, for example, of different size, you see that every single morphogen that is here depicted in different colors has a gradient. And so the cells on the outside are impacted in a different way compared to the cells in the inside by that specific morphogen. On the contrary, in 2D cultures, although, again, you have to really understand precisely, and Katja gave a great example of this just a few minutes ago, you have to really understand exactly the kind of the blueprints or really the specifics of developments at each stage of differentiation. On the flip side, once you crack that code, every cell is exposed to the same exact culture conditions. And so this is a very good way, for example, to standardize and to interrogate your platform to understand if one or more signaling pathways are necessary, are sufficient, and how they combinatorially kind of interact for the successful development of the specific cell type you're interested in. And so to basically take all these pieces together, the goal that we established in my lab, I guess, yes, probably 7 or 8 years ago, was to develop a 2D platform for in vitro generation of PGCs, of human PGCs. They're called actually PGC-LCs because they are PGC-like cells. A protocol and a platform that could be robust, efficient, reproducible, scalable, and cost-effective. And also allow at the same time for a rapid investigation of necessity and sufficiency of signaling pathways, but also the genetics of germline development, and eventually clinically translatable, because what we try to do in the lab is always, kind of envisioning a potential clinical translation of our study. And so I'm glad to to summarize 8 years of work in one slide. I think this is pretty much it. So this took the effort of 3 people over 8 years, believe it or not. Gugine started this work a long time ago, followed by by Roberta, sorry. She was a postdoc and then an instructor in my lab. And more recently, Siva from Kyle Lowe's lab has really helped bioinformatically the whole project. And in a nutshell, our protocol is 3.5 days long, and it consists of 2 windows, main windows. The first one is a window where TGF-beta and Wnt are activated, and the second one where Wnt is actively inhibited. And this is important. I will get to that in a second. And then there is, BMP that is required the first day and the third day, and ROCK inhibitor and SCF and EGF. So as I said, it took us 8 years to get here because we tried to really screen many, many more signaling pathways orthogonally. And, we finally boiled it down to the protocol that you see here. So let me start from the very end of the project, historically at least, to convince you that what we get are truly human-like primordial germ cells. And the best way to do this is really to benchmark the cells that you are getting towards or against the in vivo cell types isolated from the tissues. And so in this case, we bioinformatically benchmark the transcriptome of our PGCs against the transcriptome of fetal germ cells that were characterized and described in a couple of other papers by a Chinese group. And so what they called FGCs, which stands for fetal germ cells, they divided them. So these were isolated anywhere from week 5 to week 26, 27 approximately of gestation. The cells were taken from embryos, sorted as single cells, and then sequenced by Illumina-seq. And the authors divided the cells into 4 groups: FGC1, FGC2, 3, and 4, which basically are 4 classes bioinformatically ranked into 4 groups depending on their expression profiles. And the 4 groups basically somehow recapitulate the 3 phases of development that I showed you before. So the early phase, the germ cell commitments, and the meiotic competence. And we were very pleased to see that actually our in vitro derived PGCLCs that you see here doing an unsupervised hierarchical cluster very closely to the FGC1, which is this early mitotically active class of primordial germ cells. So you can see it here with the clustering, and you can see it here or with the Pearson correlation, where you can see that there is a fairly high correlation between the PGCLCs and the FGC1 class. This is another representation. It's a 3D Disney plot, which pretty much says the same. The next important thing that we wanted to show is that this protocol doesn't just work with one embryonic stem cells. It's very important to show that it works across many different cells. And here you see that the generation of PGCs is very robust, and we are using, in this case, a number of already described markers of PGC developments, for example, BLIMP1, SOC17, and NANOG. So you see here the immunostaining, and you see here the quantification. And what's really interesting is that, it works in embryonic stem cells, so both male and female embryonic stem cells, H1 and H9, but it also works very nicely with induced pluripotent stem cells. So here you see 3 different iPSC lines, 2 of which are male, the BJs, and 1 of which is a female iPSC line. So with this, I have a ton of other data that I don't have the time to show, but we can say that we developed a 2D platform that generates HPGCLC that are remarkably similar to fetal germ cells. And the protocol is robust, efficient, reproducible, and scalable because it can work, almost with the same consistency across many different pluripotent stem cell lines. But what's really interesting about this protocol, you remember we said that the 2D platform allows for interrogation of the genetics and interrogation of different signaling pathways. So we aimed also to show in our manuscripts that we could also show these 2 important parts or these 2 important points in our study. And so what we did was to show, for example, that Wnt modulation is necessary for PGC formation, which is a piece of novelty in the field. So here you see just a summary of the results. The protocol is divided into 2 parts, as I mentioned. And what we first have shown was, well, first of all, that the optimal Wnt activation at the beginning is just 12 hours. Not 6, not 24, but just 12 hours. And again, believe me, it took us a long time to really figure this out because we were trying all the intermediate timing of activation in between. So that's what was the first piece of information. So Wnt is necessary for 12 hours initially. But what was really striking, and it took us a long time to figure out, was that after those 12 hours, Wnt needs to be actively inhibited in order to get successful PGCs. And we got to that by doing a very simple experiment. So we compare 3 conditions. In the first one, which is called base, after the first 12 hours, we simply added media that didn't contain neither Wnt activators nor Wnt inhibitors. So there was just no Wnt whatsoever. So we were not modulating Wnt signaling. And we could get approximately 7 to 10% of PGCs. Then we tested the media where Wnt was continuously activated with shear. And in that case, we couldn't get any PGCs, as you can see here on the right. And then we tried a third media where instead of adding the activator of Wnt, we added an inhibitor of Wnt. And as you can see here, we got a very high percentage of PGCs. And so this is a very good example of how you really, in order to get in vitro differentiation of embryonic stem cells or pluripotent stem cells in general, you really need to interrogate every single signaling pathway in a very, very precise fashion. And sometimes you also have to activate it, inactivate it, activate it, inactivate it over the course of several days. And obviously this takes a lot of time to figure out. But again, what's really cool and interesting is the fact that, as you can see here, the same protocol robustly works across many different cell types, H1, H9, and in a transgenic H9 line. And again, as you can see here, 12 hours of activation are important. And I didn't show the data here, but then inhibition of Wnt afterwards is absolutely essential. And this was important for two reasons. Well, first of all, because as an example, we could really show with our platform, and this is absolutely impossible using 3D organoids because you cannot get to the same degree of intervention, because as I said, the 3D organoids being 3D do not have the same bioavailability of Wnt or Wnt inhibitors and so on across all the cells. So you cannot really do this type of interrogation studies in 3D organoids. So that was the first important thing. But the second important thing was that we could finally answer, thanks to this protocol, a long-standing question in the field, which, for the most is a very boring developmental biology question, but I have to say that I found it, very exciting, which is the following. So there was a kind of an open question in the field. So you know that the pluripotent epiblast forms a posterior epiblast. So for a long time, there was this kind of question about whether the posterior epiblast could give rise to the primitive streak first, and then the PGCs were actually derived from the primitive streak cells. While the second model was that the posterior epiblast was giving rise to, at the same time, to two distinct populations, the primitive streak and the PGCs. Now, I'm happy to report that thanks to the fact that we established this protocol, we really were able to answer this question for the first time. Well, first of all, the first thing that we observed was that the posterior epiblast was positive for OCT4, and for example, all of a sudden negative for SOX2, but was also positive for other factors, for example, NANOG, which is a pluripotency-associated marker, LIN28, EOMES, nodal, but at the same time, the posterior epiblast was also simultaneously positive for primitive streak markers, for example, Brachyury, Mixer1, and a few others. So this was kind of puzzling because there was a population that was at the same time expressing pluripotency genes and somatic genes. And then we went on and we did single cell RNA-seq of every single stage of the protocol. And to make a very long story short, we saw that in the presence again of Wnt inhibition, we could see that there was a nice, in the presence of Wnt inhibition or in the presence of Wnt activation, we were seeing a very nice separation of two populations. One population of cells that was turning on the somatic cell markers and then shutting down the pluripotency genes and keeping on the somatic markers, while another population was at the beginning expressing both the somatic and the pluripotency, and then kept expressing the pluripotency genes and shut down the somatic markers, so the primitive streak. And so again, to make a long story short, I just want to, this was again another way to confirm that NANOG, for example, was continuously expressed in a population of cells that was becoming PGCs. And this was also confirmed by immunohistochemistry. As I said, to make a long story short, we really were able to boil down the model to be the second model, the one that was successful. And so that it is not true that the pluripotent stem cells shut down the pluripotency network, and so they become primitive streak first, and then some of the primitive streaks become PGCs, but rather the pluripotent stem cells, some of the pluripotent stem cells continue to remain pluripotent and they continue to remain OCT4 and non-OCT positive, and those are the cells that become eventually primordial germ cells. And so the posterior epiblast is really capable, is a bipotent population of cells that is capable of giving rise to both the primitive streak and or the PGCs, depending in part on the activation and the modulation of Wnt, which needs to be shut down to give rise to primitive streak and needs to be, that needs to be kept active to give rise to primitive streak and needs to be shut down and inhibited to be able to give rise to the primordial germ cells. So we were very excited, believe it or not, about this. And so the last thing that remains to be done is the clinically translatable part of the protocol. Our protocol is almost fully defined, but we're still working on actually trying to make it clinically translatable. And we are about to start at some point some preclinical studies to see if this is really the case. Okay, with that, I want to, well, first of all, thank you for the attention. Obviously, the collaborators, in particular, Siva, who did a great work bioinformatically on the analysis of enormous data sets. Kyle and Lei Tang for also the collaboration. Sebastian, the funding, obviously my whole lab, it's a very nice group of people. I'm very proud of them. The funding, the institutes, and of course the departments. I'm very, very grateful to the support that I have received over the course of the last few years. And thank you all for your attention.
Thank you so much, Vittorio. It's actually really nice to see where the story has gone, and I think it shows that persistence and long-term funding really make a difference in making progress on really challenging questions. So really excited to see that. We are a little bit behind time, but we can, are there any questions in chat, Sam? So Roger has a question. We'll field one question, then we'll move on.
Hi, Vittorio. Welcome back virtually from your sabbatical presence in Europe, and really great to see you here on campus this way. I'm fascinated to know whether you regard the rejuvenating capacity of the ovary to be due down to the steroid or peptide hormones entirely, of course, and then of course those being dependent upon the germ cells, or is there something else about the ovary that has this rejuvenating capacity besides hormones?
Hi, Roger. Great point, actually. I forgot to mention that in those studies where young ovaries were transplanted into old mice, and you could see extension of longevity and extension of lifespan, health span. Remarkably, if the same studies were done with the ovaries in which the germ cells were depleted first, the extension of lifespan and the extension of health span was even more significant, suggesting that probably it's not the germ cells per se that are responsible, actually, but it's probably the somatic cells that are more important to or young granulosa cells, I should say, that are responsible for that phenotype that was or for that effect that was seen. So there is a lot of biology there to dissect and to understand, but there seems to be a kind of a dichotomy between the germ cells per se and the interaction with the somatic cells, at least in the context of longevity and health span. Yeah.
So what else could it potentially be, or what category of substances or cellular entities could it potentially be, if not steroid and peptide hormones?
It's a good question. I don't know. I don't know.
Great.
Thank you.
Thank you very much. So we'll move on. I'm pleased to introduce Dr. Gifford, who's an assistant professor of pediatric cardiology. And by courtesy, I'll let you work on getting hooked up. By courtesy of genetics, she completed her postdoctoral training in heart development and disease at the Gladstone Institute up the road at UCSF, and her doctoral in stem cell biology and epigenomics from Harvard Medical School. Recognizing that congenital heart disease is the most common congenital malformation identified in 1% of births, her research focuses on identifying the complex genetic and molecular mechanisms involved in cardiovascular development and understanding how alterations in these mechanisms lead to disease. And of particular interest, looking forward to our ongoing collaboration of understanding how congenital heart development ties in with placental development since they are connected in utero. So I look forward to your talk, and I will turn over the podium to you.
Great. Thank you, Virginia, for that introduction. We cannot see my, oh. Great. Okay, thank you. So I'll start by thanking Virginia for giving me the opportunity to tell you all about the work we're doing in my lab to understand the complex genetic mechanisms that underlie congenital heart disease and how we think that defining these mechanisms is important for precision medicine. So congenital heart disease is actually an umbrella term for lots of structural heart defects that are found in the heart at birth, with some of them listed here under this umbrella. It's found in 1% of babies. If you were to include bicuspid aortic valve, it would actually be found in about 3% of babies. So it is the most common congenital malformation identified in infants. Congenital heart disease is genetic. One of the main reasons we believe this is that it occurs in families. So if you have a child with congenital heart disease, the chances that you will have another child with this are quite high. It's variable depending on lesion. But that tells us that there's likely something going on in this family and perhaps something in the DNA in the parents of these children that's leading to the repeat incidence of this disease. Mutations that have been associated with causes of congenital heart disease have been found in a number of genes that are critical for heart development itself. And that includes NKX25, perhaps one of the first genes first associated with congenital heart disease, but it also includes additional transcription factors critical for facilitating heart development, including GATA4, NOTch1, TBX5, etc. And in my lab, we're really eager to understand the complex morphogenetic events that these transcription factors and signal transduction pathways facilitate in the context of heart development. Because we believe if we study how these transcription factors facilitate these really early stages of heart development, where you start out very soon after conception within 2 weeks with your cardiac crescent with just 2 cell types, such as the first heart field and second heart field. As you move forward in development, you have this very complex morphogenetic event that eventually leads to this 4-chambered, very complex heart. And so in my lab, we really want to understand the molecular mechanisms that dictate this process, because we think understanding this process will help us understand disease. So while we believe this disease is genetic, most genetic causes actually remain unknown. I would actually argue 95% of cases have an unknown genetic etiology. That's a little higher than what most people might tell you, because a lot of people will tell you that 25% of individuals, for example, have a copy number variant. 22q11 DeGeorge was already mentioned. So if 25% of individuals have a copy number variant, some individuals would say, well, that's it. That's the cause of disease. However, that copy number variant is not fully penetrant. So only about 50 to 70% of individuals with 22q11 have a congenital heart defect. So that variant, that lesion, is not sufficient for congenital heart disease, which is why in my pie chart here, I have it light green, because certainly that variant is contributing to the disease and to the congenital heart defect, because the risk of congenital heart disease in 22q11 is much higher than a non-22q11 population, but it is not the complete story. So that's why it's light green in my pie chart. The same is true for trisomy 21. Extra copy of chromosome 21 those individuals have a 70–80% risk for a ventricular septal defect. But again, it's not fully penetrant. So your risk is increased, but it's not sufficient. There's some other genetic, epigenetic principle or lesion or modifier that's involved. But for more than half of cases, 58% approximately, we have absolutely no idea what the cause of the defect is. And there's a number of reasons why this might be. So the first is that when you get your genome sequencing or your SNP array back for the kid that's sitting in the CVICU right now, one of the problems is that when you're prioritizing the variants identified is that we don't know all the genes important in heart development. And so if you don't know all the genes important for that organ's development, how do you know which genes to prioritize when you're looking at those that come back in these individuals? Beyond unknown genes, we certainly have not appreciated all of the variants in the non-coding genome that could contribute to disease, because it's very hard to hone in and narrow your search space in the non-coding genome and identify causal variants. The final 2 points that I'm going to touch on both today are this idea of oligogenic causes of disease, where this pie chart really has been defined using monogenic hypotheses, where the assumption is that there's a single variant that's sufficient for disease. But we know that not to be true. It's probably much more likely that there's a combination of variants that are contributing to disease. And I'm going to expand on this thought in just a second. But the other area that might be lacking, that might have been limiting our ability to move forward on defining genetic causes, is appropriate in vitro models that allow us to really dissect what we believe are causes of disease. And I'll end my talk by touching on this. So here I have a traditional effect size by frequency plot just to go over the various types of genetic mechanisms of disease. On the left side of this plot is what we consider a monogenic disease. So this is where you have a single variant with a very large effect size, and that variant is sufficient to push you above this green phenotype line. An example of this would be cystic fibrosis, right? Mutations in CFTR sufficient for cystic fibrosis. And these variants are extremely rare, right? So they're all the way on the left side of this frequency plot. They're extremely rare, if not private, so unique to an individual. The right side of this plot, however, is polygenic diseases or phenotypes. And the most famous, I would say, polygenic phenotype is height. So there are millions of variants, all with a very tiny effect size that contributes to your height. And then oligogenic is somewhere in the middle. More than one, but probably not millions. Lots of people ask me, well, how many is oligogenic? And I don't have a good answer. For me, it's more than one, but some definable number of variants that when together are sufficient for a disease, but on their own, not sufficient. And this is the scenario where you imagine you have 2 parents, they're unaffected, they have no cardiovascular problems, but then they have multiple children with disease, right? And it might be this scenario where they have variants in their genome that are not sufficient, but their children inherit that combination, and that combination is what's causing disease. So for a long time, we have thought that congenital heart disease has been on the right side of this spectrum for various reasons, and there's been some genetic associations and some computational work that's listed on the bottom right of that slide to suggest that. But it was very difficult to confirm variant involvement in multigenic analysis or multigenic hypotheses experimentally. There really were not a lot of experimental tools to test these questions. I did want to highlight, though, that this is really an ongoing topic. It's something that was just published the past couple of weeks, an update from the PCG Consortium, which actually provided really strong evidence that there are indeed rare non-coding variants out there in individuals' genomes that increase risk for congenital heart disease. So this is really the first large-scale cohort-level study that has showed that there are these rare variants that are out there in the population, and they are indeed they're enriched in individuals that have congenital heart disease. So this is a very exciting time for us in this field because we have been searching for this signal for so long. And so it's incredible to see that by combining multiple cohorts, we finally have the statistical power to ask that question. And one issue has been experimental confirmation of variants, but the other has been power, right? So when you're testing oligogenic, multigenic, polygenic hypotheses computationally, you need thousands, if not tens or hundreds of thousands of cases to do this. And so it's exciting to see that we're moving in the direction of this capability in our space, in our disease space. But so back to difficulties confirming variant involvement. So this was where we stood when I started my postdoc. But when I started my postdoc, CRISPR was coming online and the ability to really edit DNA very specifically. And that created the opportunity to test variants in very exquisite, creative experimental models. And my postdoc advisor had this case kind of in his back pocket, waiting for the day that we had the right tools to move forward. And so this is a case of left ventricular non-compaction, which I'll explain in just a minute, a familial case of left ventricular non-compaction that involved 3 children that all were diagnosed with non-compaction. They had a father that had no symptoms, but when his kids were all sick, he received an echo and they found that his heart also had this phenotype, and an unaffected mother. So, this again is why we think diseases like congenital heart disease, like LVNC, are genetic, because you have 3 children that all have this disease. So left ventricular non-compaction is thought to arise from a delay in myocardial development, because your ventricles are naturally hypertrabeculated during development to facilitate oxygen and nutrient exchange prior to coronary vasculature development. But as development moves forward, your myocardium compacts to become a much tighter, stronger muscle that can pump blood, and the trabeculi are no longer needed. So when that doesn't happen, individuals, we think, present with left ventricular non-compaction. On this slide on the left, I'm showing you a cross-section of a healthy heart, and on the right is a heart of an individual diagnosed with left ventricular non-compaction. And you'll notice that what it looks like in a human is you have this mesh-like network of tissue that's sticking out into the chamber, whereas instead, what you would hope in a healthy individual is you have this really compact, tight myocardium. So this can present isolated or complex, found with other defects. It can be asymptomatic all the way leading up to sudden death. So it's a very complicated disease. And there have been some genetic associations with this disease, but it's very complicated. And many of the genes associated with LVNC have also been associated with other cardiovascular defects. So it's a kind of a murky picture for the genetics. So our hypothesis was that this is a case of oligogenic inheritance, where some piece of this phenotype that's for all 3 of these children is explained by inheritance of some combination of variants from their parents. So we went about asking this question first by doing exome sequencing to identify non-synonymous single nucleotide variants of interest. We asked 2 hypotheses of the data to get at this question. The first is we looked for variants inherited from the father by all 3 children. We went through a somewhat traditional variant prioritization scheme, and we identified 2 single nucleotide variants of interest. The first is in the transcription factor MKL2, also known as MRTFB, so it's a myocardin-like transcription factor. This was exciting to us because it has already been implicated in epicardial development and vascular biology, and a mouse knockout of this gene has a hypertrabeculated left ventricle. So we thought, great, this is definitely going to be involved. The surprising thing from this hypothesis, though, or this test, was we also found a variant in MYH7. So I mentioned on my previous slide, this had been associated with this disease before, but the complication is that variants in MYH7, so MYH7 is a critical component of the sarcomere, they've been implicated in just about every cardiovascular disease you could define, whether it's congenital or adult. So it's a very pleiotropic gene. And so we thought that, and we've thought all along, that variants in this gene, just like in Titin or cardiac troponin T, myosin heavy chain 6, they can contribute. They're probably not sufficient. So we still have it on our list of interests because it's likely involved in the phenotype, but again, not sufficient for the phenotype. And it wouldn't explain why the father is asymptomatic until imaging, and the 3 children all have symptomatic disease and require therapeutic intervention. So then the second hypothesis we asked of the data was whether we could identify genetic modifiers that are inherited from their asymptomatic, perfectly healthy mother by all 3 children. The slight switch we made in our prioritization approach here was that we kind of loosened the deleteriousness and the computational predictions on these variants. We didn't apply this prediction to this hypothesis because you wouldn't necessarily expect a damaging variant if it's a modifier. If it was really damaging, very deleterious, you might expect the mother to be symptomatic. That was our hypothesis, at least. And so when we applied this scheme, we identified a single nucleotide variant in the transcription factor NKX25, critical for heart development. This was especially interesting because this is out in the population around 1 in 1,000 people. So this is the perfect modifier, right? It's out there in the population, and only in combination with other variants would it lead to disease. It's not enough on its own. And so this was our hypothesis, that these 3 variants, when inherited together, would lead to a phenotype such as that seen in this family. So I'm going to summarize my entire postdoc in one slide. And I'm just going to share with you one part of that, which is the mouse work, which was only made possible because of CRISPR-Cas9 gene editing. So we generated mice that carry the 3 orthologous variants, and then we asked the million-dollar question: do triple heterozygote animals have abnormalities in the left ventricular wall? I'm not going to call it LVNC because LVNC is a human phenotype, and I'm not here to argue about whether what we see in mice is the same thing we're going to see in humans, but at least do we see something that resembles what you might see in humans? And so when we look at 4-chamber views of the mouse heart, we indeed see increase in hypertrabeculation in the triple heterozygote animals. So you can see a wild type animal here with the left ventricle on the right, relatively smooth myocardium, especially down here in the apex. As you increase variants, including in the NKX25, you'll see that there you start to see this red signal, which is a stain with an endocardial protein, and you can see the trabeculi jutting out into the lumen of the ventricle. Same with this double mutant here, which represents the father's genotype, trabeculation. And then you move over to this triple heterozygote, and again, you see these trabeculi into the left ventricle, and it goes all the way down into the apex, which is a traditional view of LVNC in humans. And so this was really exciting to us that we were indeed able to recapitulate these protrusions into the left ventricle in these triple heterozygote animals. We, of course, looked at this over many, many litters over a long period of time, and we were able to see what I showed you as representative images before, that apical thickness goes down as you add mutations in these animals, so the apical myocardium is thinner. And then the level of trabeculation as quantified using fractal geometry increases, meaning the complexity of the left ventricular space, that red signal, is increasing as you add more variants. So with this and a lot of additional work, we felt very confident that this was perhaps the first experimental evidence that a human congenital or structural defect was in fact oncogenic. So this was very exciting, but it's one family, and it was my entire postdoc. And so really when I was starting my lab, the question is, how do we go from this mouse work and a little bit of iPS cell work that takes a lot of time and a lot of money to something that actually will work for precision medicine, right? We need to be able to solve cases much faster in a much more robust way than you making a new mouse even though, you can make a mouse in a couple of months now, but that's still not going to be fast enough to make it feasible for precision medicine. So when I started my lab, I thought a lot about how can we create a robust, scalable human system that would allow us to study the time points and cell states that are involved in disease when they're perturbed. And I thought a lot about these specific questions because of what we know about heart development and when heart development happens during embryonic development and all the cell types and complexities and morphogenetic events that are involved. So this was some single-cell RNA-seq from a gastrulating human embryo that was published a couple of years ago. And I thought it was really interesting and unexpected because what it actually showed, so this is the single-cell RNA-seq of the entire dissected embryo, and all of the cell types that they were able to identify already at this gastrulation, roughly Carnegie stage 7 embryos, they already have what they think are cell types representing advanced mesoderm. So already this very early stage of human development, they're getting cell types that you would probably find in a heart. And so here on the right side of this slide, I'm focusing in on the mesoderm. And then on the very right side is some genes that we identify as being expressed in heart-related cell types like cardiomyocytes. And so what you can see is there's already MYL7 and cardiac troponin T. So genes expressed in cardiomyocytes are already expressed in the embryo at this very early stage of development. So what this tells me is that if we want to understand why congenital heart disease is happening, this is the stage of development that we need to study. And not only that, But we need to be studying how all of these cell types develop coincidentally, because in this embryo, they're all developing right next to each other. They're relying on paracrine signaling. There's communication going back and forth. And so you also need a system that allows you to tease apart that signaling axis if you want to understand how its perturbation might lead to disease. So what we've been really focused on in my lab for the past year or so is developing and adapting a cardiac organoid approach, because we think that this system represents a human multicellular model of differentiation. And so we've adapted a protocol that was published from Sasha Menjin's lab, which involves taking human pluripotent stem cells, you aggregate them in a U-bottom plate, you modulate all of our favorite signal transduction pathways, including Wnt, TGF-beta, retinoic acid, and BMP signaling. And then after about 8 to 10 days, you get this multicellular cardiac organoid that includes multiple cell types we think you would find in the heart. So this was our question. This is a published protocol. Can we adapt this and make this work in our lab? And the answer is indeed yes. So by day 8, we have beating cardiac organoids, which is seen here. And then on the right is an example in the GFP channel. We have an MYL7 GFP reporter, which represents cardiomyocytes. And for the remainder of my talk, I may actually call them cardioids. That's our short term for a heart organoid. We refer to them as cardioids. So we hope that we have many cell types that represent cells you might find in the heart. We hope that's the hypothesis and based on previous literature. So in order to look into this a little bit more, we of course did single cell RNA sequencing. I'm just going to share with you a tiny sliver of information from this data, but this is 10x single cell RNA sequencing from days 2 through 12 of differentiation. It's many samples integrated, a little over 37,000 cells. And we do, in fact, have cardiomyocytes, as you would expect. We also have what we believe is endocardium based on expression of genes such as endomucin and MPR3. We also have very specific smooth muscle cell types. So PENK is expressed specifically in outflow tract smooth muscle. So we think we have that cell type. And then perhaps most surprisingly is we think we have cardiac neural crest based on expression of genes such as TFAP2A, TFAP2C, the DLXs. So surprising, but very interesting and exciting to us, because the cardiac neural crest is actually critical for heart development. It migrates into the neural tube, and its perturbation is actually associated with a number of congenital heart defects. So then the question is, can we use this system to actually go in and understand differentiation in disease? And this is what I'm going to show you next, a close collaboration with Ayal, who is a postdoc in Will Greenleaf's lab. So we wanted to do a pilot study and look into a gene that we would expect would have a phenotype. And we chose myocardin. So myocardin is a transcription factor that's critical for smooth muscle development. It is lethal by E10-5 for its smooth muscle reliance. It eventually, later on in development, also prevents cardiomyocyte differentiation. And you can see that in our data, it is indeed expressed in cardiomyocytes. We used a line that Ayal developed that allows DOX-inducible CRISPR interference to knock down myocardin. And we see that there's no expression of myocardin, or very little expression of myocardin at days 5 and 8. And you can also see in the myocardin knockdown, we are not getting MYL7 positive cardioids. So it seems that this is doing what we would expect. We then used single-cell RNA-seq to understand what was happening in these myocardin knockdown cardioids. And what we indeed were able to confirm is that there's no cardiomyocytes present. So our wild-type cardiomyocytes found at day 10 are here in green. The myocardin knockdown cells in day 10 are these purple cells, and they're in a different place in this UMAP. So they're expressing different genes. They have a different transcriptional profile. So this confirms what we would probably expect, that myocardin is important in cardiomyocyte differentiation. The other really exciting result is that we actually saw a difference over here in this cluster at day 10 in the cardiac neural crest. And so this was perhaps expected because myocardin has been implicated in neural crest-derived smooth muscle development. And so it seems like perhaps that's what we're modeling here. So we can just zoom in really quick, and I'll show you that when you look at these clusters, it looks like the control is basically going in one direction, and there's this bifurcation where the myocardin-derived cells are going in a different direction. When you look at the genes that are differentially expressed, a lot of them look like they're related to migration. So one of the hypotheses is that perhaps myocardin is important for neural crest migration. And this is a really exciting opportunity for us to define more genes and regulatory elements that are perturbed in this knockdown that might be involved in cardiac neural crest migration, for example. And then the final analysis I'll share with you is something that Matteo brought up earlier. actually. So what we did was use cell chat and known ligand receptor pair expression to figure out which cell types talk to each other and which signaling axes are perturbed in the myocardial knockdown. And so what we found is that in the wild type setting, if you just focus on the colors that are not grayed out, in the wild type setting, the neural crest is signaling to the endocardium through the SEMA3 pathway, the SEMA3A pathway. But in the knockdown cardiac neural crest, you can see the thickness of this line, it's much thinner. So the hypothesis is that the cardiac neural crest in the myocardial knockdown cannot signal to the endocardium. And you can see that here, this is showing you the UMAP, where you can see that SEMA3C at least is expressed highly in the wild type, but not in the knockdown. And this is exciting because perturbation of this signaling axis has already been implicated in outflow tract septation. So we actually think that this might represent a really cool model for understanding genes and regulatory elements that are involved in outflow tract septation. And what I didn't tell you is that this is actually multi-omic data. So we have both shared single-cell RNA-seq as well as ATAC-seq for this experiment, so we can actually go in, for example, and look at regulatory elements that are impacted. So we think we have this system that might be a really great way to understand the genetics of congenital heart disease. And I just wanted to end by saying that in my lab, we're also very interested in the comorbidities associated with congenital heart disease, like placental abnormalities, and also the neurodevelopmental delay component, but in particular, this genetic aspect. So something that we're wondering, because the heart is essentially a vascular organ, is there vasculature that's in the heart and also in the placenta that might share mechanistic information, might share genetic information? So if you have perturbations to the vasculature in the heart, maybe those perturbations and those mutations are also contributing to some of the lesions that you might see in the placenta. And this is something that we're very eager to think more about. In order to do that, I just wanted to give shameless plug for two groups that I'm part of. We are recruiting patients at LPCH for a couple of different studies to try and collect as many samples along these lines as possible. The first is the Man Charities Biobank that I started through a very generous donation. And so we're recruiting individuals specifically that have congenital heart disease and their families. I'm happy to talk to more people about that if they're interested. And then I'm also part of a broader NIH consortium called Gregor, and we're interested in recruiting any individual with any rare disease for various genetic studies. So if you're interested in either, please let me know. So very quick take-home messages. Most cases of congenital heart disease have no known genetic etiology. They remain to be defined, and that's really something we're interested in. There's evidence that it's multigenic, both experimental, now that we can do some mouse experiments and some organoid experiments, as well as some genetic associations. And we believe that iPS cell models, especially organoid models, might be a really good way to try and tease apart some of these unknown genetic etiologies. So I'll stop there and just acknowledge every single person in my lab that has contributed to this project. This has really required a huge team effort. So everyone listed here has contributed in some way. I did point out an experiment, this myocardial experiment that was done by Ayel, as well as Jackson, who's an RA in my lab, my collaborators, and I specifically want to acknowledge all of the patients and their families that contribute to doing this research, because without them, we wouldn't know where to look in the genome. So with that, I'll take questions if there's time. Thank you for your attention.
Great, thank you very much. Are there any questions in the chat? Any questions? So I actually had a question regarding thinking about your pie chart, and the trisomy and CNV that you think need another genetic component to lead to disease. I was curious as to why you think it might be genetic versus environmental, i.e., it's the gene environment interaction in utero that leads to the disease rather than all genetic.
Yeah, I think it's possible. I do think, for example, diabetes would be a good another sort of more environmental component. That could be part of it. I certainly think that, but I think some of it might also be genetic. But that's a very interesting question, and I think there are some models that might allow you to tease apart, for example, diabetic models of the complication, but it could be any of them. So
actually, that's utilizing your cardioid with environmental triggers.
Yeah, it's very
Might
possible.
be a very interesting
Yeah, and that's
path to
something
go.
that James Priest was doing before he left. So I was sad that he left, but that would be great if we could get that model to work. So Deepak's done a little bit of that in his lab, so I'm trying to stay away from that for now. But yeah, it's a great idea.
Yeah, and then I was really intrigued with the family that started you
Yeah.
on this adventure.
Yeah.
To have all 3 children have both the maternal and paternal inheritance.
Yeah.
Kind of goes against almost Mendelian odds.
Well,
and so I was curious whether, do you think there's some aspect to explain that?
so you might think that. However, if you look at all the inherited variants of these 3 children, there's actually hundreds of variants that they all collectively inherited. So it's not that rare. We actually, so I don't think it's biased. I don't think that there was biased inheritance of either of those variants that would go against, this traditional Mendelian approach. I just think there's more variants out there than we think. And so it turns out there was still a lot we had to sift through. It wasn't like there was only 3 or only 10. There were hundreds. And those are the ones that passed filters and QC, right? So I just think there's more variants out there than you would imagine. So it's unlucky for sure, but more common than you would think. Yep.
So really enjoyed your talk. I was just wanted to offer a comment about the gene environment interaction, because I work with an organization in India where they've consulted on about 200,000 children in the last 5 years for congenital heart disease. And a large majority of those mothers have anemia during pregnancy. And in fact, we found clusters of villages where up to 5% of children are born with one single congenital heart defect, like tetralogy of Fallot.
Okay.
So that's. I'm sorry, this is boring to someone. I truly think you should subject your cardioids to, changing iron levels, for example, or other, kinds of things.
Yeah, I think that's a great point, and I would be happy if I had more people in my lab to explore that. But I would actually counter and say that, something I'm very interested in is genetic ancestry and how your genetic background contributes to phenotype penetrance. And so it is possible that those villages have rare variants that are more common there than in other populations. So what I would love is to be able to go to those villages and collect some samples to do some DNA sequencing for our biobank. Because I think that's another angle to it. But maybe it's the combination, right? Maybe it's that they have some variant that combined with the anemia is sufficient for this complex genetic mechanism, right? Whereas someone without that variant, the anemia is not sufficient, right? Yeah.
We have a biobank with samples from all 200,000.
Oh, great. Perfect. Let's talk. Okay.
All right. Let's thank Dr. Gifford again. And what a beautiful example of why we wanted to have this symposium, was to bring people together and have exactly that wonderful exchange occur.